AS Level Biology 9700, Paper 2, Variant 1, October November 2024. Question number 5 and 6. Question number 5 is trypsin is an enzyme which catalyzes the hydrolysis of casein, a protein found in milk. Milk that contains casein had a cloudy white appearance. As the casein is hydrolyzed by trypsin, the milk changes in appearance to a clear, transparent, colorless solution. A student carried out an experiment to investigate the effect of enzyme concentration on the rate at which trypsin hydrolyzes casein. The student added a solution of trypsin to a sample of milk and recorded the time taken for the milk to become transparent. The student repeated the experiment with different concentrations of trypsin. All other variables were kept constant. So the figure 5.1 shows the results from the experiment when the concentration of trypsin increases from 2 to 4%. The time taken for the milk to become transparent decreases by 48%. So calculate the percentage decrease in the time taken for milk to become transparent when the concentration of trypsin increases from 0.25 to 0.5. So write your answer to the nearest whole number. Nearest whole number. So we are going, first of all, we will move to the figure 5.1. So this is the figure 5.1 in which they have shown a graph, and according in this graph we can so here you can see this graph, and according to this graph we have to find out the values on the percentage concentration of trypsin, which is 3.1, and it is should be 0.5 and 0.25. So we will draw, first of all we will draw at 0.25. So you can see it will be drawn in this way at 0.5. The value will be very difficult to draw the line, straight line actually. You can see, you can draw with the help of scale. Okay, and the second line will be drawn on 0.5. So this is the actual 0.5. It will be in this way. You can draw with the help of scale. I'm drawing on screen. So this is the reason the line is not very proper, but you will draw it with the help of scale. So the values are between 350 and the other value, which is at 0.25, it is 350, and the value at 5 is 230. So in this way, now we will make use of these values to calculate the percentage decrease. We will make use of this formula. So we are going back to the space where we have to use the formula to calculate the percentage decrease. Now we know the values. The initial value is 350 because uh, it was decreased from 0.5 to 0.25. So the initial value is, or initial time you can see, it is 350 and final is 230. So we will make use of percentage decrease is equal to initial time minus final time divided by initial time multiplied by 100. So we will make use of this formula. We will put the values. Percentage decrease is equal to 350 minus 230 divided by 350 multiplied by 100. So it is equal to 120 by 350 into 100, and it is 34%. So the answer after calculation, the answer is 34%. So it was all about the percentage decrease, that is it is 35 to 230, and it is about 34%. Now the next question is explain the results. In figure 5.1 shows that as the concentration of the trypsin increases, the as the concentration of the trypsin is increases, the time for, first of all, the time taken for milk to become transparent is decreasing. So we will discuss these results, that how it is taking place, that how this time is decreasing in this case. So you know that in this figure, in figure 5.1, the graph in the start, the graph was around, the value was about 390 or less than 400, then it was decreased, and its value was decreased. So it means that the as the concentration of trypsin is increasing, the time taken for milk to become transparent is decreasing. So this means the rate of casein hydrolysis increases with increasing trypsin concentration. So we can say that enzymes work by binding to their substrate at an active site. So when the enzyme concentration is low, there are fewer enzyme molecules available to bind with the substrate, limiting the reaction rate. So as the enzyme concentration increases, there are more enzyme molecules available to bind with the substrate, leading to a faster reaction rate, because the substrate will always have to bind with the active site. So when there will be more active sites or with a higher trypsin concentration, we can say there are more active sites available to break down casein molecules simultaneously. So that this speed up the process of hydrolysis, resulting in the milk becoming transparent more quickly. So within the range shown in graph, the rate of the reaction is roughly proportional to the enzyme concentration. However, it's important to note that this proportionality usually does not contribute indefinitely. So at high enzyme concentration, the reaction rate may plateau because other factors like become limiting. So uh, after this explanation, I would like to explain the answer which I have written. I have this. The result in figure 5.1 shows that as the concentration of trypsin increases, the time taken for the milk to become transparent decreases. As you have seen in the graph, that the graph is, you can say, in this way. On this side, they had taken the concentration of trypsin of enzyme, you can see that, and the other side, the time taken to hydrolyze. So in this way, the time is decreasing in this way. So this shows that a higher concentration of trypsin speeds up the hydrolysis of cancer, leading to faster clearing of milk. Because already I have shown you that if the concentration of the enzyme is high, it means there are more active sites available at that time. So more, it will be less time will be taken to hydrolyze. So leading to faster clearing of milk, this happens because enzyme concentration affects the rate of reaction. So it will increase the rate of reaction. So at higher trypsin concentrations, more active sites for substrate binding, increasing the rate of casein breakdown. However, the curve suggests that the effect levels off at high concentration, likely because the substrate becomes the limiting factor. So if the substrate is not there, if the concentration of substrate is decreasing, then there will be no, you can say, effect on the rate of reaction. But when there is availability in the active sites and substrate molecules are also available there, the rate of reaction will be very fast. So it was all about this question. And next question is, trypsin has the potential to be used in a wide range of industrial processes. The use of immobilized enzyme in industrial processes has many advantages. Scientists investigated the effect of temperature on the activity of trypsin immobilized on the surface of a material and trypsin free in solution. So shows the results of the investigation and temperature at 0 degrees centigrade. So the percentage of maximum activity of immobilized trypsin and percentage of maximum activity of trypsin in free in solution. And with the from this table, you can see as the temperature is increasing from 25 to 65, the percentage of maximum activity of immobilized trypsin is also increasing from 60 to 100%. On the other hand, you can see the third column in which the percentage of maximum activity of trypsin free in solution, the activity of this is decreasing from 100 to 5. So you can say as temperature is increasing, the activity of immobilized trypsin is increasing, and the trypsin, which is free in solution, its activity is decreasing. It shows that the immobilized enzymes can work better at high temperature because this is the ability by which they can withstand with high temperature. So you know that these enzymes are biological catalysts to speed up the chemical reaction living organisms. So in industrial processes, using enzymes can be very beneficial, but free enzymes can be unstable and difficult to recover. Immobilization so is a technique where enzymes are attached to an inert insoluble material. So this provides several advantages to them. So in this way, the enzymes are attached to a solid sport. They can be easily separated from the reaction mixture and reused multiple times, reducing cost. So after this explanation of the table, I would like to read this question. State a reason for the difference in percentage of maximum activity of immobilized trypsin and trypsin free in solution at 25 degrees C. So you can see over here the process of immobilization might cause slight changes in the enzyme's three-dimensional structure, or sometimes you can see that it also changed the specificity, uh, or you can say sometimes it can cause a little bit change. And in this way, but the other enzyme, which is freely available in solution, can be denatured at high temperature. As you know, the optimum temperature for enzyme extraction is 35 degrees centigrade to 40 degrees centigrade. So between this temperature, you can see the enzyme become denatured. But when immobilization has been occurred, so due to immobilization, the movement and flexibility are restricted, and due to which the enzyme's ability to bind to its substrate uh, is enhanced, so it becomes fast. So in this way, I would like to say the process of immobilization might cause slight change in the enzyme's three-dimensional structure, which could affect its active site and reduce its catalytic efficiency at 25 degree centigrade. And then next question is suggest and explain why the percentage of maximum activity of immobilized trypsin at 55 is higher than in the percentage of the maximum activity of trypsin free in solution at 55 degrees centigrade. Already explained, that is the at 55 degrees centigrade and 25 degrees centigrade. There is change, you can say, in the activity. You can see over there at 25 degrees centigrade, the percentage of maximum activity of immobilized trypsin is 60%, and percentage of maximum activity of trypsin free in solution is 100%. Whereas at 55 degrees centigrade, the percentage of maximum activity of immobilized trypsin is 95%, and the percentage of maximum activity of trypsin is 20%. So it means that the enzyme work best in uh, at its optimum temperature when it is free in solution. But when it is immobilized, so its activity is increased. So its efficiency is increased. It is not denatured at the temperature, at high temperature. So enzymes are protein, and high temperature can cause them to denature. Immobilization often enhances the thermal stability of enzymes, because immobilization enhances their stability. So the sport material can restrict the enzyme's flexibility, making it more resistant to heat. So at 55 degrees centigrade, free trypsin is likely experiencing significant denaturation to lose their shape, whereas the immobilized trypsin maintains the shape of active sites even at 55 degrees centigrade. So we can say that this temperature immobilization is very helpful to save the enzymes from wastage and from denaturation. And in this way, we can also increase the activity, increase the efficiency and activity of the enzymes. So it was all about question number 5. Now, question number 6. Question number 6 is there. You can see figure 6.1 shows a plant cell in a stage of mitosis. And uh, So some of the substrates shown in figure 6.1 contains DNA. Use a line labeled D on figure 6.1 to indicate one of these structures. Name the stage of the mitosis shown in figure 6.1. You can see this picture and you can see the chromosomes are just start moving from the equator towards the opposite poles. So we can say that this stage is anaphase because after when they are in the center, it means they are present at the equator, and when they are at the equator, it means the phase is metaphase. Now they have started moving towards the opposite pole, so it means the stage is anaphase. Next question. Okay, this is the DNA. Use a line labeled D on figure to indicate one of these structures. Now, next is colchicine is a chemical used by scientists to study mitosis. The chemical inhibits the organization of the microtubules in prophase of mitosis. So the cell shown in figure 6.1 had not been treated with colchicine. Explain the evidence in figure 6.1 that shows the cell had not been treated with colchicine. So you can see colchicine will inhibit the organization of the microtubules in the prophase of mitosis. So here you can see the ester fibers are also there around the centrioles. Centrioles are present on the opposite poles. You can see these spindle fibers which help the chromosomes to move towards the opposite poles. So it means spindle fibers are there, microtubules are there, and these chromosomes are attached with these uh, microtubules or the spindle fibers. So I would like to say that the presence of organized spindle fibers indicates that the microtubule formation was not inhibited, suggesting the absence of colchicine. It means colchicine was not there. So the alignment of chromosomes at the metaphase plate suggests that spindle fibers are functional and that the cell was not treated with colchicine. So we can say that this is the way that the colchicine was not used for this purpose. Now, the answer after this explanation, the answer which I have written, explain the evidence in figure 6.1 shows that the cell has not been treated with colchicine because the organized spindle fibers are visible, which indicates that microtubules formation was not inhibited. So the alignment of chromosomes at the metaphase stage suggests that the spindle fibers are functional and the cell was not treated with the chemicals. Thus, the cell is observed in state which is beyond the state where colchicine would arrest mitosis. So that was prophase in which the colchicine can inhibit the activity, but they have shown the metaphase and after metaphase they have just started the anaphase. So the chromosomes are moving towards the opposite poles. So whereas the chromosomes are moving apart near to reach the poles, so we can say that there was no use of colchicine in this photograph. So it was all about question number 4 and 5. Please like, share, and subscribe my channel. And thanks for watching.